This is B.Maheswaran, Assistant Professor, Department of Environmental Science, Virudhunagar Nadu College, Virudhunagar. In this video tutorial, we are going to learn about effects of xenobiotics. Xenobiotics produce a wide range of biological effects which including pharmacological responses, toxicity, immunological responses and cancers. When xenobiotics is a drug compound, then phase 1 reaction which results in the activation or termination the actions of daughter compound cells produced from the phase 1 reactions. If a drug is a pharmacologically active in the body prior metabolism, it passed into phase 2 reactions. So even a low amount of or low concentrations of toxic xenobiotic compounds are also very dangerous one. For example, cyanide. So cyanide is a chemical compound in which it is a xenobiotic. It comes under the category of xenobiotics. Because a very low amount of cyanide, it will be more poisonous nature, right? So you all know that. So cyanide is a very dangerous one. So which results into death. So nonetheless, there are some xenobiotics like drug molecules which is administered sufficiently amount to that the body do not exert toxic effects. First, cytotoxicity. Right, so xenobiotics injure cells by means of many mechanisms. Right, so cytotoxicity is nothing but a cell injury. Right, so the one considered here is a covalent binding to the cell macromolecules of reactive species of xenobiotic compounds produced by the metabolite. These macromolecules target the DNA, RNA and protein macromolecule also. And second one is a reactive species, right? So the reactive species of xenobiotics bind to the protein macromolecule. Its antigenicity, altering its antigenicity property, right? So that is captain is a very well-known example for antigenicity. Right, so captain is a very small molecule. It's an incomplete antigen. So the xenobiotic is said to be act as a captain, that is small molecule. Already I know, I told that that by itself doesn't stimulate any antibody synthesis at all, but it is combined with the antibody once formed. On the other hand, the carrier doesn't perform any immune response by itself, right? So it doesn't elicit any immune response by itself. Once the protein molecule is bind with the carrier molecule, then it perform immune responses. Okay, so here it is a captain molecules, so it's a small thing, right? So this is a carrier molecule. It may be a protein which combines and forms complete antigenic protein, right? So this is a captain, this is carrier molecule, it forms complete antigen. Okay, so drugs like penicillins are captain. So penicillin is a very known example for captain molecule. The resulting antibody which can damage the cell by some other immunological responses that totally disturb the cellular mechanisms by means of biochemical pathways. And third reaction of activated species of chemical carcinogens with the DNA molecule which are considered to be a great significant in chemical carcinogenesis. For an example, benzene alpha pyrene. So benzene alpha pyrene is the activation by the enzyme so-called monooxygenases in the endoplasmic reticulum of the cell to become carcinogens. That's why it's called as indirect carcinogenesis, right? So here it's the need of monooxygenase enzyme. to the cell. Okay. Epoxide hydrolyse. This is an enzyme which of interest because it can exert a protective effect against some carcinogenic agents. Products from the actions of monoxygenases on some carcinogen substrates are called epoxides. They are highly poisonous, and as well as they are maybe mutagenic, carcinogenic, or both, right? So like cytochrome P450 epoxide hydrolyse, which is also present in the membranes of endoplasmic reticulum of a cell, right? So which act on these compounds, converting to them into much lesser active dihydrolyse. Though the reaction which is catalyzed by epoxide hydrolyse, which can be represented as the mentioned equation. So here epoxide is converted into dihydrodyl by means of epoxide hydrolyse. Thank you.